Hello, everyone. Thank you for joining this episode of our Discover Molecular Interactions webinar series. My name is Vivian, and I am responsible for scientific communication at Dynamic Biosensors. And each month, we explore different biophysical assays, and we discuss how they can be used to measure molecular interactions. And we also present different application examples from our collaborators and customers, and also internal R&D. In case you have not seen the update on our event website, unfortunately, our guest speaker of today, Eva, had to cancel on health grounds, but I will fill in for her and describe the essay that she was going to explain. And she kindly also agreed that I can show some of her data, so we can offer you almost the same content for this episode as planned. And I am also supported today by Anaï. She is one of our application specialists, and she has vast experiences with the enzymatic activity assays. So I'm very happy that she's here today. Hi, Anaï.
Hi, Vivian. Thank you for the invite.
Now, if you have any questions during the talk, please feel free to use the Q&A tool, or if you can't see that in the app, you can also use the normal chat function, or we can also unmute you after the talk and speak directly. And now, without any further ado, I will jump right in, and we will talk today about catalytic rates of nucleic acid modifying enzymes, for example, reverse transcriptase activity. And I would like to start by quickly give an introduction on why we should at all care about nucleic acid modifying enzymes and why it is interesting to look into their catalytic activity. So nucleic acid modifying enzymes have a very diverse range of roles, not only in biology, but also in disease and research. For the biological roles, there are functions in genome replication or gene expression regulation, but in disease, they also are very important, for example, in the case of viral infections or gene expression dysregulation. And then there's, of course, also the role of nucleic acid enzymes in research, because many of those enzymes are already used as an important tool for molecular biology. For example, if we think about polymerases being used for PCRs. So all of these things make it very obvious that this is a highly investigated topic, and the current assay is mostly based on gels, which is pretty tedious and also not a real-time assay. But what we would like to have is an assay that investigates something that looks approximately like this. So we have, for example, a primer strand, a polymerase that comes in and that elongates the primer strand according to a complementary template strand. And we want to be able to measure this process not only rapidly and reliably, but also to look at it in real time and not only as an endpoint analysis. And then, so to speak, as the cherry on top, we would also like to be able to separate the association of the enzyme to the template strand from the activity, so the actual synthesis of the nucleic acid strand. The tool we suggest to use for this is an assay that is based on the switchSENSE technology. So this video shows how we just translated what we want to observe onto a chip surface. And of course, this video has a lot of information in it, so let's deconvolute this a bit and look at it step by step. At first, I wanted to show you the Helix adapter chip. Our chips have a microfluidic channel with two different measurement spots that are gold electrodes. And on each of these spots, we have these anchor strands. And to these anchor strands, you can then immobilize a DNA nanolever. The nanolever consists of an adapter strand, which also carries the fluorescent dye. This is very important for our method of detection. And then we have also a ligand strand, which can carry any ligand of interest, for example, a protein or also the RNA or DNA sequence. Now, in a normal binding assay, this would be our nanolever setup, and we would then flow in the analyte in the microfluidic channel. And upon binding, this would then change the local environment of the fluorescent dye, leading to a change in fluorescence intensity. If we want to use this assay format now for enzymatic activity assays, we need to tweak it a little bit and make some tiny detailed changes. So the setup would then look approximately like this. We now have a primer on the adapter strand, and we have a template strand that carries the fluorescent dye. Let's have a look at what this would look like if we have no enzyme present and also no substrates, I mean DNTPs present. In that case, we would have our primer strand and we would have the template strand, which is now in a single-stranded state. So that makes it pretty wobbly. So it will most of the time be very close to the gold surface of the electrodes. And the gold will quench the fluorescence intensity of the attached dye. Now, if we now go on and add the nucleic acid modifying enzyme, for example, a polymerase, as well as the substrates like the nucleotide, the antibodies, we will now see the elongation of the complementary strand, and this will lead to the synthesis of a now double-stranded molecule. The double-stranded sequence is much more rigid than the single-stranded one, so now the distance of the dye from the chip surface will increase and the fluorescence intensity will also increase. And this is how we can measure the catalytic activity of the enzyme in real time by observing this increase in fluorescence intensity. So now we can put all these things together again and have a second look at the video I quickly showed you at the beginning of the talk. So we can see that the enzyme comes in, binds to the primer, we add the nucleotides, and then the complementary strand is synthesized, and the dye is increasingly coming away from the quenching surface. And we see this as an increase in the fluorescence signal as the dye is increasingly away from the surface. Now, I told you in the beginning that our chip has two spots. We have a second spot that we can use as a real-time reference, and there we can, for example, put a double-stranded sequence from the beginning, so we have a baseline signal that will show the full fluorescence all the time. The workflow would look approximately like this. You start with an empty chip surface, only the anchor strands are present. Then you functionalize the surface with these nanolevers containing the primer and the template strand. And then you inject the enzyme. And in this step, you can look at the association of the enzyme. In the next step, you then inject the substrate, so the NTPs, so you can look at the elongation and the catalytic activity of the enzyme. Now, the very important thing about this workflow is, as you can see, we separately look at the enzyme association and the enzyme activity. And this is very convenient also if you want to look at inhibitors and their effect on specific nucleic acid modifying enzymes. Because in a workflow like this, you can add an inhibitor during the enzyme association step, and you can look at its effect on the enzyme binding to the template. And you can also add the inhibitor during the activity step, and you can separately look at the effect of the inhibitor on the catalytic activity. I will also show an example of what the data for these inhibitor assays could look like later on. But first, I would like to start with the example by Eva about the reverse transcriptase activity. So the project of Eva was examining unnatural base pairs. Unnatural base pairs are very interesting because they augment the chemical diversity of sequences, and this is very interesting for the generation of, for example, new aptamers or also for new catalytic nucleic acids. And Eva looked especially at the unnatural base pair of TPT3 and NAM, and she wanted to investigate whether reverse transcriptases are able to retain these unnatural base pairs during the process of reverse transcription. She used a plethora of methods to investigate this, but for this talk, I will mainly focus on the kinetic measurements. So at first, the setup of the assay was mostly like I just described to you. And you can see here the fluorescence traces for the activity of two different reverse transcriptases that were investigated. We have the MMLV reverse transcriptase, and then we have an engineered version of the MMLV that is called superscript 4. And you can see here that with increasing concentrations, from bottom to top, we have increasing concentrations of NTPs, and you can see that over time, as the reverse transcriptase synthesizes the cDNA, we will see an increase in fluorescence intensity. And we can derive from this an observed weight, and if we plot this against the concentration, we can derive a K-cut, and you can directly see that the engineered version of the reverse transcriptase has a higher catalytic activity compared to the original enzyme. Now, the question was whether these reverse transcriptases are capable of retaining unnatural base pairs during the process. And there are several scenarios that we can imagine when the enzyme comes across an unnatural base pair modification in the sequence. So one option would be that the enzyme is just skipping the modified base pair and causes a mismatch. Another option would be that the enzyme is processing the unnatural base pair and includes the complementary unnatural base. And the third option would be that the process is stalled and the transcription ends at the point of the modification. To investigate the capability of the enzyme and to investigate what of these scenarios is happening, 4 different nanolever setups were used. So on the left, we see the normal template strand without any modifications. This is the setup I have shown you before in the comparison of the 2 enzymes. Then we have 2 setups with 1 or with 2 modified bases in the template strand. And finally, we have 1 control setup. The control nanolever is shorter and its length corresponds exactly to the length of the other nanolevers until the point of the modifications. So with this, we can look for the scenario where the reverse transcription is stopped at the point of the modifications. So let's have a look at the data. At first, only normal DNTPs were injected, so we do not have the complementary base of the TPT3 in the mix here. And you can see that for the normal unmodified template strand, we see what we have seen before, the elongation of the full template. For the modified template strands, we now see a much lower amplitude. So that means that not the full template is elongated by the reverse transcriptase, as is the case in the first example. And we can also compare this with our shorter control nanolever, where we can see that the amplitude is matching. So that means that the transcription process probably only is performed until the point of this modification. Now, in the next step, we added now the complementary base to the TPT3, the NAM. Here you can see the same picture for the normal unmodified strand. However, in the strands with the modifications, we still see a very low amplitude that corresponds to the amplitude of the shorter control strand. So from this, we can conclude that the MMLV reverse transcriptase seems to be not able to process this unnatural base pair and is stalled at the point of modifications, causing a shorter elongation of the template strand and not a full transcription of the full template. Now I would like to turn to another application example that is, as I have promised in the beginning of the talk, the inhibition of enzymatic activity. And for this, we looked at a polymerase. So here we make use of the specific feature that we can differentiate between the binding and the activity of the enzyme. So we first looked at the association of the polymerase to the primer sequence in presence of an inhibitor. We can see that the association of the enzyme will change the local environment of the fluorescent dye and will cause a decrease in fluorescence intensity. Now, with increasing concentrations of the inhibitor, this signal doesn't change at all. So that means there's no effect of the inhibitor on the association of the enzyme. Now, in the next injection step, we injected the nucleotides as well as the inhibitor, and we can see that with increasing concentrations of the inhibitor, we also see a decreased catalytic activity of the polymerase. We can also plot this decrease in activity over the inhibitor concentration and derive an IC50 value from this. So this would be one example where you can screen an inhibitor for its specific effect, whether it's occurring at the association step or at the activity step of the process. And I also have another enzyme as an example. This is a helicase. This is a very interesting example because it's sort of the other way around. In this type of experiment, we do not have the activity of the enzyme that synthesizes the strand, but instead it will unwind an already double-stranded starting sequence. We can also look at the association of the enzyme, and we also see a decrease in fluorescence intensity here, similar to the polymerase association. Now, for the activity signal, we will see the signal the other way around. So if we add the substrate, in this case it is ATP, in increasing concentrations, we will see a decrease in fluorescence. That is because in this case, the strands will be unwound, so the fluorescent dye will come closer to the surface again once the sequence is single-stranded, and we will see a decrease in fluorescence. And we can also plot this as an observed rate over the concentration and derive different constants from this, like the kcat. So this is already the end of my presentation. I hope I could give you a good overview of what is possible with the analysis of enzymatic activity in this system with this technology. And I hope that I could also briefly show you some of the data that Eva was going to show. I also recommend reading her publication on this that is already published, and I am open, and Anai is also open to answer all your questions. And thank you for your attention. So we already have a question. How compatible is this with evaluating reverse transcriptase extension and processivity through secondary structure oligos? Did we ever try this with an oligo with a secondary structure?
Not really, but I wouldn't think that there is a problem with that. In terms of the assay, it would look exactly the same, just that the the template strand would have the secondary structure, and whether this secondary structure is stable or too stable, one will need to try if the enzyme is able to actually open this secondary structure and continue polymerizing the strand. Yeah, one would need to try that.
We're always happy to try on new projects, so please also feel free to reach out and we can organize a demo or just try out your samples. By the way, if you were interested in secondary structures of nucleic acids, we also have a different assay for that where we actually use 2 fluorescent dyes, and then we can visualize the change in secondary structure of the nucleic acid, because once the nucleic acid folds in a specific way, this might cause a thread between the 2 dyes. So we can also send you some details on that if you're interested. Okay, the next question would be, what is the length of RNA or DNA that is required for the assay? And what distance change is required for the shift from quenching of the fluorescent signal?
So we have a kit for the people that is not interested in having like sequence specificity. We have a kit where the template is 32 nucleotides. So this is enough to actually see a nice fluorescence increase, but the template can be also longer. I think we have tried up to around 50 nucleotides for the template, and that still works very nicely. One needs to take into account that after, so this linearity of the increase of the fluorescence compared to the distance towards the surface, it's around 100 nanometers. So after that, if the template is longer, we might not be able to see like a nice linear increase of the fluorescence. And can you repeat the second question, please?
What distance change we need to see the unquenching of the fluorescent dye?
What distance change? I think this is also related to what I just mentioned. So the distances that we are able to differentiate are between lower than 100, 150 nanometers in length. So I think everything around that, it's it would be possible. Of course, very small changes of, let's say, 1, 2 nanometers, 3 nanometers, that is not resolvable, but you would see like a change whenever the distance or the building of the bases is more.
Mm-hmm. And I think the nanoliver also already has a minimum length, so to say. So like we have already just keep here, we already have the anchor strand. So it will already have a specific distance from the chip. So I think minimum length is also not a problem because we already have like the standard part of the nanoliver, right?
So this standard part is 96 nucleotides. So this is already the minimal length from the chip surface.
Okay. If there are no more questions, I will just go back to the last slide. And I invite you all to send us a message or reach out to us on our website if you're interested in more information or if you would like to test your specific system with our technology, and check out our website for upcoming webinar episodes. We also have the recording of the previous episodes. So for example, if you're interested in nucleic acid binders, there was also a very nice episode about multi-domain proteins binding to RNA. That could be something you're interested in. So we have the recordings of those on our website too. And thank you for joining today. Thank you for your questions. And I hope to see some of you in an upcoming episode. And I wish you a very nice rest of your day. And thank you, Anai, for joining us and answering the questions.